The opening fact that changes everything. In 1962, scientists froze bull sperm in liquid nitrogen at -196 degrees Celsius. 40 years later, they thawed it and successfully produced a calf. Cells can survive indefinitely in liquid nitrogen if you freeze them correctly. Today you will learn exactly how that works, and why getting it wrong is catastrophically irreversible. Cryopreservation is your insurance policy. Every cell line you build, every primary culture you painstakingly establish, every genetically modified clone you spend months creating, all of it is one incubator failure, one contamination event, or one bad batch of media away from being gone forever. Unless you froze it properly first. The physics of survival at -196 degrees Celsius. At -196 degrees Celsius, the temperature of liquid nitrogen, all biochemical reactions essentially stop. Enzymes do not function, metabolism does not occur, DNA does not replicate. TME, for a cell in liquid nitrogen, does not pass in any biologically meaningful sense. A cell properly stored in liquid nitrogen in 1990 is, biologically, exactly the same age as when it was frozen. This is remarkable, but it comes with a critical caveat. The journey from 37 degrees Celsius to -196 degrees Celsius is not biologically neutral. It is a minefield. The cells that survive the thaw are only the ones whose journey through that minefield was precisely controlled. Every experiment you run depends on the quality of the cells you use. Cells at high passage number accumulate genetic changes. Cells that have been through stress change their biology. Cryopreservation lets you hit save on your cells at their optimal state, so every experiment starts from the same verified, authenticated baseline. Without this skill, you are always building on sand. The physics of freezing, why cells die and how we stop it. Before you touch a cryovial, you need to understand the physics. Cryopreservation is not simply put cells in the freezer. The physical processes of freezing are actively hostile to cell survival. Every decision in the protocol, the cryoprotectant, the cooling rate, the thawing speed, is a direct response to a specific physical threat. Threat 1. Ice crystal formation. When water freezes, it expands and forms crystalline structures. Inside a cell, this means that the same water that normally flows freely through membranes and around organelles transforms into rigid, expanding, jagged ice crystals that puncture membranes, rupture organelles, and destroy the cell from within. This is why fast freezing, just throwing your cells into a -80 degrees Celsius freezer without any protocol, kills nearly all of them. The rapid temperature drop causes rapid ice crystallization throughout the cell. By the time the cell reaches storage temperature, it has essentially been shredded from the inside. The Goldilocks problem: too fast. Massive intracellular ice crystal formation. Cell destroyed from within. Too slow without DMSO. Cells dehydrate as extracellular ice forms, drawing water out by osmosis. The remaining intracellular solutes reach toxic concentrations, called solution effect injury. Just right. Approximately -1 degree C per minute, which allows gradual dehydration while preventing lethal ice formation inside the cell. The hero, DMSO, dimethyl sulfoxide. DMSO is a small, membrane-permeable organic solvent that enters cells with remarkable efficiency. Once inside, it remains liquid at temperatures where water would crystallize. It replaces intracellular water, lowers the freezing point of the cell interior, disrupts ice lattice nucleation, and maintains membrane fluidity. All at the same time. Critical safety warning: DMSO toxicity. DMSO is toxic to living cells at physiological temperatures. At 37 degrees Celsius or room temperature, DMSO rapidly damages cells. The same membrane permeability that makes it a great cryoprotectant makes it a cellular toxin when cells are metabolically active. You have approximately 15 to 20 minutes from adding DMSO to your cell suspension before significant toxicity begins. Work fast. Keep everything cold, 0 to 4 degrees Celsius. Do not let your cells sit in DMSO at room temperature. The standard DMSO concentration, 10%. 10% DMSO balances cryoprotection versus toxicity, the universal mammalian standard. Lower concentration, inadequate protection, more ice crystal damage. Higher concentration, more toxicity during preparation and thawing. Thawing, the most dangerous step. This surprises students. Thawing is where most cells die because small ice crystals undergo recrystallization if warming is too slow. Small crystals merge into large, lethal ones, DMSO toxicity rises with every second above -20 degrees Celsius, and osmotic shock from concentrated cryoprotectant meeting normal media can lyse cells if dilution is too sudden. The golden rule of thawing. Warm fast in a 37 degrees Celsius water bath. A 1 milliliter cryovule should go from -196 degrees Celsius to fully thawed in 60 to 90 seconds of gentle agitation. The moment the last ice crystal disappears, stop. Transfer immediately. Dilute the DMSO immediately. Every second of delay costs viability. STR profiling, genetic fingerprint of your cell line. Short tandem repeat. STR profiling is the genetic authentication of your cell line. Before banking any cell line, send a sample for STR profiling and match against ATCC or DSMZ reference. Misidentified cell lines have been found in an estimated 15 to 20% of published biomedical research. STR profiling is cheap insurance against being one of those papers. Have you ever wondered why cells can still die during slow freezing, even when no ice crystals form inside them? The answer is something called solution effect injury. Here's what happens. As freezing begins, ice forms first outside the cell, but ice crystals are made almost entirely of pure water. That means salts, proteins, and other dissolved molecules get pushed out of the ice and trapped in the remaining liquid around the cell. And suddenly, that surrounding liquid becomes extremely concentrated, or hypertonic. Now the environment outside the cell is saltier than the inside. So by osmosis, water starts rushing out of the cell. The cell begins to shrink. It dehydrates. Its internal chemistry becomes dangerously concentrated. Inside the cell, proteins start unfolding and denaturing. Membranes become stressed and damaged. Metabolic balance collapses. So even without sharp ice crystals stabbing the cell can still die from extreme dehydration and chemical stress. This is why slow freezing without cryoprotectants is often fatal. And this is where DMSO becomes critical. DMSO penetrates directly into the cell. It replaces part of the intracellular water and reduces the osmotic imbalance between the inside and outside of the cell. That means less water is pulled out during freezing. The cell stays more stable. Proteins remain functional. Membranes stay protected. So DMSO is not just preventing ice crystals from forming. It is also preventing the cellular equivalent of drinking seawater until you implode. And that's why cryopreservation is really a balance between two dangers: too much ice formation, or too much dehydration. DMSO helps cells survive both. The complete cryopreservation protocol. Freeze-to-Thaw. No lab access? No problem. These protocols give you the exact decision points, quantities, and timing. Read them until they feel automatic, because when working with DMSO and liquid nitrogen, there is no time to look things up. Part A. Preparing to freeze. Pre-freeze checklist. Cells at 70 to 80% confluency. Phase contrast checked. Healthy morphology confirmed. Passage number documented. Cryovules labeled. Mr. Frosty filled with fresh isopropanol, equilibrated at room temperature. Freeze medium prepared and chilled to 4 degrees Celsius. Timer ready. Do not begin without all of these. Step 1. Prepare freeze medium, FBS first, DMSO last, always. Always add DMSO to the FBS, never the reverse. DMSO mixing is exothermic, it releases heat. Adding large volume FBS to DMSO spikes temperature. FBS first, DMSO second. Then chill to 4 degrees Celsius before cells ever touch it. Step 2. Harvest your cells. 1. Remove media. Wash with PBS, 2x for sensitive lines, 1x for robust. 2. Add trypsin, 0.25%, 2 milliliters per T25, 3 milliliters per T75. 3. Incubate 3 to 5 minutes at 37 degrees Celsius. Check every 90 seconds for detachment. 4. Neutralize immediately with 2 to 3x volume complete media on detachment. 5. Transfer to 15 milliliters conical, centrifuge 300 grams by 5 minutes. 6. Remove all supernatant, residual media dilutes DMSO concentration. 7. Resuspend in cold freeze medium. Target 1 to 3 by 10 to the power of 6 viable cells per milliliter. Step 3. Count, dilute, and label cryovules. Use trypin blue exclusion or automated counter. Target 1 by 10 to the power of 6 viable cells per milliliter per vial. Every vial needs cell line name, passage number, freeze date, operator initials, cell concentration, freeze medium composition, and bank type, MCB or WCB. Step 4. Load Mr. Frosty and freeze 8. Ensure isopropanol is fresh and filled to the marked line. 9. Place labeled cryovules in the rack, close lid. 10. Transfer immediately to -80 degrees Celsius freezer. 11. Do not open freezer during 12 to 16 hour cooling period. 12. After overnight, transfer vials to liquid nitrogen for permanent storage. Liquid nitrogen safety, non-negotiable. Always wear cryogenic gloves, not regular lab gloves, face shield, lab coat. LN2 contact causes immediate cryogenic burns. Rapid vaporization in enclosed spaces displaces oxygen, unconsciousness without warning. Never seal LN2 in a closed container. Work in ventilated areas. Know your oxygen depletion monitor. Part B. Thawing protocol. Speed is viability 13. Prepare before retrieving vial. Warm complete media to 37 degrees Celsius. Label of 15 milliliters conical. Water bath at 37 degrees Celsius. Timer ready. 14. Retrieve vial with cryogenic gloves and forceps, never bare hands. 15. Immerse in 37 degrees Celsius water bath. Agitate gently, do not fully submerge cap. 16. Watch for last ice crystal to disappear, 60 to 90 seconds. Remove immediately. 17. Wipe exterior with 70% ethanol before opening. 18. Transfer dropwise into 10 milliliters pre-warmed complete media, prevents osmotic shock. 19. Centrifuge 300 grams by 5 minutes. Remove DMSO containing supernatant carefully. 20. Resuspend in pre-warmed media, transfer to appropriate flask. 21. Incubate 24 hours. Trypin blue viability check before any experiment. Post-thaw viability standard. Target. Greater than 70% viable, ideally greater than 85%. Count using trypin blue exclusion in a hemocytometer. If viability is below 70%, do not proceed with experiments. Let cells recover 2 to 3 passages and re-evaluate. Proceeding with compromised cells does not save time, it guarantees the experiment will need repeating. Imagine this. You remove a cryovule from liquid nitrogen, drop it into a 37-degree water bath, and then walk away to make tea. 5 minutes later, the vial is finally thawed. What's the problem? The problem is DMSO toxicity. At cryogenic temperatures, cells are essentially frozen in time. Their metabolism is shut down, and in that inactive state, DMSO is relatively safe. But the moment the cells begin warming, everything changes. Metabolism starts turning back on. Cell membranes become active again. Proteins begin functioning. Ion gradients restart. And now the cells are alive enough to be damaged. Because DMSO is protective during freezing, but toxic at warm temperatures. That means the longer cells sit thawed in DMSO, the more damage accumulates. A proper thaw should take only 60 to 90 seconds. But if thawing stretches to 5 full minutes, and then sits even longer before dilution, cell viability can crash dramatically. What could have been 85% viable cells may drop to 40% or less. And the worst part? The damage is often invisible at first. The cells may still attach. They may even look normal for a few hours. But later, growth slows. Stress responses activate. Experiments fail. Data becomes unreliable. This is why thawing is not a passive step. It is a race against DMSO toxicity. Warm fast. Remove immediately when the last ice crystal disappears. Dilute the DMSO right away. And remember, make the tea after the cells are in the centrifuge. The cells cannot wait. The tea can. 5 mistakes that destroy cell banks, and how to avoid every one. These errors have ended careers, wasted millions in research funding, and set entire research programs back by years. They are all preventable. Read each one, internalize the fix, and never make them. Mistake 1. Freezing cells at high passage number. After 8 months of growing HeLa cells to passage 47, you decide to freeze some, just in case. Problem. At passage 47, your HeLa cells have accumulated dozens of mutations relative to passage 5. Different gene expression, different drug responses, different biology. You are banking the wrong cells. The fix. Freeze as early as possible, ideally within 5 to 10 passages of receiving cells. Master bank at the lowest possible passage. Every passage is selection pressure and potential genetic drift. Bank early, bank often, bank low. Mistake 2. Adding DMSO directly to the cell suspension. You prepare cells first, then add DMSO directly to the tube. DMSO mixing is exothermic, it generates heat. Your metabolically active cells are now in contact with warming DMSO. Toxicity begins immediately. By the time you aliquot into cryovules, viability is already compromised. The fix: prepare freeze medium first, separately from cells. Mix FBS and DMSO, FBS first. Chill to 4 degrees Celsius. Only when freeze medium is cold and ready do you harvest and resuspend your cells. Minimize DMSO exposure time. Mistake 3. Slow thawing. Cryovule in the water bath. You get distracted, come back 10 minutes later. Cells have been marinating in increasingly toxic DMSO for an extended period. Post-thaw viability, 20%. You did not lose those cells to bad freezing. You lost them to bad thawing. The fix: stay at the water bath. Watch the vial. The moment the last ice crystal disappears, remove it, wipe with ethanol, open, and proceed immediately. 60 to 90 seconds. This is not a step you walk away from. Mistake 4. Not testing viability after thawing. You thaw, replate, and proceed to your experiment the next day. Results are irreproducible and 3 days of work are wasted. The problem: post-thaw viability was 35%. Your starting conditions were undefined and cell biology was already compromised from the moment of plating. The fix: always trip and blue a sample before replating. Below 70%. Do not proceed with experiments. Establish the culture, recover for 2 to 3 passages, re-evaluate. Proceeding with compromised cells does not save time. It guarantees repetition. Mistake 5. Storing all vials in one liquid nitrogen tank. Your LN2 tank develops a slow leak over a weekend. By Monday, the LN2 level has dropped below your cryovules. Cells thaw partially, refreeze, thaw again. Recrystallization destroys everything. Your master bank, working bank, every backup, gone. No recovery option. The fix: never store master bank and working bank in the same tank. Ideally, different buildings or facilities. Modern tanks have electronic monitoring that alerts when levels drop, worth the investment. A 2 a.m. tank alarm is infinitely better than a Monday morning catastrophe. A researcher freezes cells at passage 40, adds DMSO directly into the cell suspension, and then stores all 30 vials in a single liquid nitrogen tank. Question: which mistake is causing the most immediate damage right now? Pause for a second and think about it. The answer is adding DMSO directly to the cell suspension. Why? Because that damage is happening immediately, in real time. DMSO is not harmless at room temperature. The moment it touches metabolically active cells, toxicity begins. And there's another problem: mixing DMSO directly into the cells generates heat. The reaction is exothermic, so now your cells are exposed to warming DMSO, while still fully metabolically active. That combination is dangerous. Every second the cells sit there, viability is dropping. By the time all 30 cryovules are aliquoted and capped, a large fraction of the cells may already be compromised. Now, the other 2 mistakes are absolutely serious. Freezing cells at passage 40 means you may already be working with genetically drifted cells. Different mutations, different gene expression, different biology. That damages the scientific reliability of your future experiments. In storing every vial in one LN2 tank? That is a catastrophic risk. One tank failure, one leak, one weekend without monitoring, and your entire cell bank could disappear permanently. But those problems are future problems. The DMSO mistake is happening now. The cells are being damaged while you are standing at the bench. That's why proper cryopreservation is all about timing and preparation. Prepare freeze medium first, mix FBS first, DMSO second. Chill it to 4 degrees Celsius. Only then should your cells ever touch DMSO. Because in cryopreservation, small timing mistakes become massive viability losses. Cryopreservation at scale, the infrastructure of biomedical science. What you learned in this episode is the foundational infrastructure of all biomedical science. Every time a researcher buys a cell line from a TCC, opens a vial from their master bank, or accesses samples from a clinical biobank, they are relying on exactly the science you now understand. The major cell repositories. These repositories authenticate cells using STR profiling, verify Mycoplasma free status, maintain passage-controlled master banks, and distribute at defined low passage numbers. ATCC has been operating since 1925. When you buy from them, you are buying from the world's most experienced cryopreservation institution. Clinical and Research Biobanks. UK Biobank. 500,000 participant samples, blood, urine, saliva, stored at -80 degrees Celsius and LN2. NCI Biorepositories. Millions of cancer patient tissue samples supporting oncology research globally. Cord blood banks. Hematopoietic stem cells from umbilical cord blood, cryopreserved at birth, used for bone marrow transplants. iPSC banks. Reprogrammed stem cells from disease patients. Any researcher worldwide can order and use them. Reproductive medicine. Sperm, oocytes, and embryos cryopreserved using the same physics you learned today. The Reproducibility Foundation. One of the biggest crises in biomedical science is irreproducibility. A significant contributor is the use of misidentified, overpassaged, or contaminated cell lines. Researchers who use authenticated, low-passage cells from reputable repositories, properly cryopreserved and banked, produce dramatically more reproducible results. The skills in this episode are not just technique, they are the foundation of scientific integrity. A researcher receives a cell line from a collaborator. No documentation, no authentication report, no contamination testing, just a flask with cells inside. Question: what should they do first before starting experiments? The answer is: never begin experiments with unauthenticated cells. Step 1 is always Mycoplasma testing. Why? Because Mycoplasma contamination is one of the most dangerous hidden problems in cell culture. The cells may still look normal under the microscope, but internally, everything changes. Gene expression shifts, metabolism changes, growth rates become abnormal, drug responses become unreliable, and worse, Mycoplasma spreads silently to every culture it contacts. One contaminated flask can compromise an entire laboratory. So before anything else, test for Mycoplasma. Second, perform STR profiling. STR profiling is the genetic fingerprint of the cell line. It confirms that the cells are actually what the label claims they are. Because throughout scientific history, thousands of studies have been performed on the wrong cell lines. Researchers thought they were studying breast cancer, but were actually growing melanoma cells. Or thought they had one patient-derived line, when it was completely replaced by another. That means years of experiments, papers, drug studies, and conclusions were built on the wrong biology. Third, once the cells are confirmed clean and correctly identified, establish a master bank at the lowest possible passage number. That master bank becomes your protected reference stock. Then create a working bank for routine experiments. And only after all of that, should experimental work begin. Because an experiment performed on contaminated or misidentified cells does not just waste time. It can generate false conclusions that spread through the scientific literature and harm the entire field. Now, let's get into the summary of this episode, the 3 pillars of cryopreservation. First, DMSO prevents ice crystals, but is toxic to live cells at room temperature. DMSO is the hero and the villain of cryopreservation. At -196 degrees Celsius, it prevents ice crystal formation and solution effect injury, making long-term survival possible. At 37 degrees Celsius, it penetrates cells and disrupts membranes with the same efficiency. You have 15 to 20 minutes of working time after adding DMSO. Keep everything cold, work fast, dilute immediately after thawing. Second, slow freeze, fast thaw, always. The -1 degree Celsius per minute cooling rate from Mr. Frosty gives cells time to dehydrate safely without intracellular ice formation. The thawing rate should be as fast as possible, 60 to 90 seconds in a 37 degrees Celsius water bath. These 2 rules are non-negotiable. Every deviation reduces viability. Slow thawing is not gentler, it is deadly. Third, authenticate and bank every cell line before starting experiments. STR profiling is your genetic verification. Mycoplasma testing is your contamination clearance. A master bank at low passage is your insurance. A working bank is your operational supply. None of these steps are optional if you want reproducible science. An experiment built on unauthenticated, overpassaged, or contaminated cells is a building built on sand. And with that, we complete Module 1. In the next episode, we are moving into Module 2, Mammalian Cell Culture. We'll explore the famous cell lines that built modern biology. HeLa, HEK293, and the rest of the gang. Where did these cells come from? Why are they so important? And why does almost every biomedical laboratory in the world use them? That's coming up in the next episode. If you found this episode useful, don't forget to subscribe our channel. Don't forget to like, share, and leave the comment below. We'll see you in the next episode. Thank you for watching.